Good morning, ladies and gentlemen. My name is Christian Zeine. I am the product manager for pharmaceutical reference materials, and I welcome you to the second part of the webinar today. We talked about 1, 3, 5, and 7. We talked about this already 2 weeks ago. Today we will talk about the truth number 2 and 4 and number 6 here. Our truth number 2 is that an incompletely characterized reference standard bear risks of hidden costs. And the following slides that I show you, they are about case studies that come courtesy from my colleagues in Luckenwalde from the LGC Germany GmbH, where we produce our impurity reference standards, and my colleague Dr. Andreas Zeine provided those slides to me. We had a couple of years ago we had a customer who complained about the N-oxide of metoclopramide. It did not correspond to his expectations that he had. We provided that material to him, and he said there is a mistake in it because it does not match the material or the infrared spectrum of the material that he used before. We looked first of all onto the synthesis of the impurity standard that we made and characterized. And you can see that we start from the drug substance itself, from the API itself, and because you can oxidize both amine groups here, so we did a protection of the one at the aromatic ring to be sure that we can oxidize just this one here. And after the protection of the aromatic amine group, we then oxidize this one here, and after successful oxidation we hydrolyze the material back to the N-oxide itself. To that regard, we could be quite sure that we have the right material at hand. We also looked then into all the identity data once again, and you can see here that this compound is also described already in the literature. You see here literature results for the shifts of the NMR signals, and you can see here the shifts of the signals of our material, and you can see that these both are quite similar. So to that regard, we could have been quite sure that we had the right material at hand. We also looked into the CNMR spectrum, but again we could explain the structure with the position of the signals, of the shifts of the CNMR signals here, and it was in conjunction and consistent with the structure that we had here as well. We also looked on mass spectrometry data again, so you can see here the main peaks at 316, that is the molecule peak plus hydrogen. We could explain the one here at 271 and at 227 and at 184, also with this fragmentation here. We also had a look on the infrared spectrum of our material, of course also on the one from the customer. We come back to that in the next slide, but here's the one from our compound, from our material, and the characteristic bands and supposed assignments you can see here, and all the main bands here which are necessary to explain. We could explain with the structure once again of the substance. What we did then as well was that we made an elemental analysis and that confirmed also our expectations. You can see it here. You have the result here of our material, the CHN analysis of our material, and here you can see the specified value, the theoretical value that comes up, and you see a very good correlation here between these results. We were also thinking maybe for some reason we might have the hydrochloride at hand, but it did not correspond, the hydrochloride salt, but it did not correspond really to what is theoretically available for the hydrochlorides. You see, they doesn't match at all, so they match much better with the free base values than the hydrochloride values here. And also we performed a chloride test, a simple chloride test with silver nitrate. This one also went negative. We evaluated then once again the customer spectrum as well, and what we saw and what we missed in our spectrum were typical bands of an ammonium salt that were shown in the one from the customer. And an ammonium salt that is forming, of course, when you have a hydrochloride salt at hand. So the spectrum that the customer showed or provided to us showed these ammonium salt bands, very much comparable to what you can see also when you have the API as an hydrochloride at hand. So what our suggestion was and our suspicion was that the customer had the hydrochloride at hand and was thinking all the time he was having the free base. And the customer then himself performed a chloride test, which went then positive. And so he confirmed then back to us that we were right in that case and that he had the hydrochloride at hand without knowing it for the time he used it. This is not so much a problem when you want to use such a material for identification only, for peak identification in an HPLC, because in the HPLC, in the mobile phase or in the solvent that you use for your analyte, you normally split the salt up, and in the chromatogram it in the end it will show as the free base peak, so you don't see any problems there. But when you want to use it quantitatively and you think all the time you have the free base at hand, but instead of that you would have the hydrochloride salt or a sulfate salt or a hydrate itself without knowing, then of course you can have issues, because then you might overestimate or underestimate the analyte in your real sample, of course. And using a hydrochloride without knowing, it's happening quite often. We have a lot of queries on different IR spectra that we can explain with this phenomenon. The case number 2 here to show you about that is for a trazodone impurity, for this one here, chlorophenyl piperazine hydrochloride. That is a compound that we wanted to purchase from fine chemical trade, for example, in order to characterize it and to make a reference standard out of that fine chemical then. So we looked into the literature and found their data for the free base again, for a monohydrochloride and for a dihydrochloride as well, with this data here. And then we looked also into the fine chemical trade, what is available for ordering. And we looked at 3 suppliers. Supplier number 1, he provided in his catalog, he provided the molecular mass of the free base, he provided the CAS number from the hydrochloride, and he provided also the melting point of the hydrochloride. But we saw then that it was the dihydrochloride that this supplier was providing. The second supplier we looked at, and we looked onto the catalog data, there once again the information we got from him in his catalog, and all the data here, they point out to the hydrochloride, as you can see, for molecular mass, for the CAS number, and also for the melting point. But also here, this supplier, he was actually providing the dihydrochloride rather than the hydrochloride itself. Supplier number 3, when we looked at his data in his catalog materials, he was providing the molecular mass of the hydrochloride. He was providing the CAS number, however, of the dihydrochloride, as you can see here, and also the melting point pointed to the dihydrochloride. But when we analyzed this one here, in fact, it turned out that this is really the hydrochloride that we wanted to make a reference standard from. So in the end, we used this material here, characterized it, of course, thoroughly once again, in order to provide maximum information to our customers there. So let's see, what can you learn from this truth number 2? So what I really want to tell you is, be careful about your sources of reference standards. When there are identity issues, for example, as we have seen, not recognizing salt forms, that can cause you serious problems that you may not be aware of for some time. And as I said, it's not so much a problem if you really want to use it for peak identification purposes, but when you want to use them quantitatively, it might pose problems for you. Because such an incomplete quantitative analysis, for example, you think you have the free base and instead of that you really have the hydrochloride lying in front of you, such a incomplete identity analysis will give you problems when you do quantitative analysis later and will lead to standards, will lead to reference standards that are overestimated in their assay figure, and in turn it will also lead then to overestimation of impurities in your products with all the consequences later on. And in the first presentation about the 7 truths, in the truth number 1, we talked about toxicological studies for impurities that are above qualification threshold, and that can easily take you 3 or 4 months longer to bring the substance to market than expected. It is also a wise approach to not rely just on the chromatographic purity alone. Again, this is just an incomplete quantitative analysis characterization that you would do. Because if you just use chromatographic purity and do not look on water and residue solvents that are normally, those things are normally not discovered by checking on the chromatographic purity, then you are missing several % there on water and maybe also on residue solvents. And as we have seen in the case study number 2, just the slide before, it's recommendable not to just procure fine chemicals and take their reference standard just for granted. The best thing to do is to use dedicated reference standards from trusted sources, and we ourselves from LGC Standards, of course, we consider ourselves as such a trusted source. I would like to show you now what we have here for you about truth number 4, and that is one that you might have experienced already as well, that the pharmacopoeial monographs can change, and so can also change the supply of impurity reference standards that are used for that monographs. And such monographs from the United States Pharmacopoeia and the European Pharmacopoeia, for example, they are constantly developing due to technical progress, for example, but also sometimes or very often also to regulatory changes, of course. When they change, when the monographs change, at the same time the pharmacopoeias accommodate their very often also their sourcing issues for impurities. And you will see that very often that over the years and over the change of the monographs, also the unit sizes of impurity reference standards from the pharmacopoeias are changing. And normally they are changing in a way that less material is provided per unit size from the pharmacopoeias. What they also sometimes do is they switch from neat materials to materials that they call then drug substance XYZ for peak identification or for system suitability. That is all good to work with the monograph because that is what they are made for, but there are a lot of users who use the materials from the pharmacopoeias also for purposes apart from the monograph. But what can happen is that the monograph is changing and then that the reference standard for the impurity is disappearing and you do not have that availability anymore for that material. So what I want to tell you here is it is also again recommendable not to specify explicitly a USP or an EP impurity reference standard in your dossier. Okay, so and I would like to show you a few examples on the next slides what I mean with that, and that is what we are doing right now here. So I had to look on the catalog number 40, for example, from the European Pharmacopoeia. That was issued in 2004. And what I did there was I compared it to material that is now, right at the moment, actually available. And I looked at the first 25 reference standards from that catalog, and I looked then into the newest catalog, what is available right now. Here are the examples of the first 25 reference standards. So you can see here that for isobutylol impurity I, from 20 milligrams of neat material, this changed to a unit size of just 4/1000 of a milligram lyophilized material. So you cannot do much more with that than just one check according to the monograph. Aceclophenag impurity F, this one changed in unit size from 50 milligrams to 20 milligrams. Aceclophenag impurity H, that was downsized from 25 milligrams to 10 milligrams. You can see that here. And aciclovir impurity A, that was available in 2004 as a neat material with a unit size of 30 milligrams. That one was completely withdrawn and was replaced by peak identification and system suitability mixtures. So in such a case here, if you would have written into your dossier that you want to use this special impurity here from the European Pharmacopoeia, and then it is not available anymore, that is a situation you do not want to find yourself in. And if you would extrapolate the finding for the first 25 reference standards here, when you would extrapolate that over the whole EP range in the year 2004 and would look on it right now, then you would find that 198, so close to 200 impurity standards are now available with only a diminished unit size, and you would also find that 66 impurity standards would have been completely withdrawn. This numbers here, of course, is just an extrapolation, and it's also connected to a high uncertainty, of course, but nonetheless it is some figures here, some numbers that you might want to think about. That was already truth number 4. We are looking now into the truth number 6, in that impurity descriptions from the pharmacopoeias will not necessarily match each other. And I just before we started the webinar, I had another customer with whom I needed to explain and discuss the whole thing once again. What is happening very often is that a customer is complaining that the purchased impurity standard is not matching his expectations. And when we are calling then that customer and following up that complaint, it turns out very often that what the customer wanted initially was a certain impurity from the European Pharmacopoeia impurity list. So it might be that the customer wanted a certain impurity here, or that he wanted a certain EP or USP impurity that was not, at that time, was not on stock at the pharmacopoeial warehouses. And what the customer then decides for himself was that he assumed, and sometimes that is a wrong assumption, as it can be the case that the impurity description of EP and of USP is matching each other, but it is also very often possible, and you should always look it up, it is always also possible that the description is not matching each other, and that this would be a wrong assumption to do so. So, and if he is then thinking the impurity A of API XYZ is corresponding to the related compound A of the same API, then of course issues can come up when you do the analysis here. And the next slides, I show you a few examples for that. For example, the acetaminophen, or as we call it in Europe, the paracetamol related compound A from the USP is not identical with impurity A from the European Pharmacopoeia, but is identical only with paracetamol impurity H from the European Pharmacopoeia. Another example is the ibuprofen related compound C from the USP. That one is identical with the ibuprofen impurity E from the European Pharmacopoeia. And the last example here is that the omeprazole related compound A from USP is identical with the omeprazole impurity D from the European Pharmacopoeia, and not with impurity A from the Pharmacopoeia, the European Pharmacopoeia. So our recommendation here is, when you are in need for a certain impurity reference standard, and when you are unsure which product is the best for you, it is always advisable to contact us either via your local office or via this email here, askus@lgcpharma.com. Okay, that was already the end of the presentation. I would like to close with some closing remarks. LGC Standards is part of the LGC group, and our Science and Technology section at LGC is acting as the National Metrology Institute for chemical and biochemical measurements in the United Kingdom. And to that regard, we are comparable, for example, to the NIST in the United States. We manufacture pharmaceutical reference materials, and for APIs and impurities we have over 3,000 standards available, and they are prepared in this site, at this site here, which is in Luckenwalde, situated in Luckenwalde in the south of Berlin, Germany, where we produce all those materials for the pharmaceutical organic compounds. We also have metal impurity materials available. They are suitable for ICP, OES and MS applications. We manufacture those in our premises in the United States of America. And our sister company there is the company VHG, which is providing materials for especially these applications and services for these applications as well. We have constantly new developments, and we are happy to announce that in October 2013, during the CPHI here in Frankfurt in Germany, we will launch a range of primary reference standards. We are ISO 34 and ISO 17025 accredited in most laboratory sites. We have also contract services available for custom synthesis, for custom analysis, for characterization of customer material, and also for customized packaging and storage. And that I would like to explain you in the next slides very quickly what I mean with that. Because what you can think of is when you want to set up a reference standard, you go through every one of these steps here, and each one is a single step in itself and has a start and an end. And what we are offering to our customers is that they also can book each one of this step here. So we can base each of these steps on either on internal quality standards that the customer are giving us, or to our quality standards 17 or 25 or ISO guide 34. We can also do it under GMP conditions as well. And we have also supplementary processes available after setting up the reference standard for ongoing stability monitoring and retesting there as well. So you can see here two services or two examples what we do for two of the world top 10 pharmaceutical companies. So you can see that the one company here, the upper company here, is sourcing and doing an initial quality control on their own, and then they provide to us their raw material, as we call it, and we characterize it and certify it and then package it. Sometimes we package it even before we do characterization and certification. Sometimes we do that before, store the material in bulk, and then if a customer is ordering, an internal customer from one of those companies, we package them and distribute those materials. You can see here the second company is doing all the analytical work still in-house, but they say, look, the distribution we really want to use you for, and the storage as well. So please store the materials for us, and once an order is coming in from one of our internal customers, please package it and distribute it to every site in the world. We also do services over the whole chain. We do that for a German medical company, for a German subsidiary of the world top 10 pharma companies, and also for one of the world top 3 generic companies. We provide such services here. And you can see that here in that slide here, an example. For example, we set up a primary and secondary standard for itraconazole for one of our customers. And you can see here that it was requested by the customer that we bottle 45 units of that material. And those costs came up here, and that was an average price per unit. The cost per unit was then resulting in 153 euros. This is still a lot of money, but you can easily see that the longer you let such a cooperation run and the more units you would need, the less expensive the unit price would become. You can see that in the second example where we set up a controlled material for another customer, for toluidine hydrochloride here. You can see here that 126 bottles were ordered, and you see that already the unit price is going down to 69 euros here. We set up a primary standard for Etomidate here. The customer let us bottle 300 units single-use vials, for example, and you see here then that the price per unit goes down to 26 euros here. To use the single-use vial makes much sense. You open it, you use the material, and then you discard the rest, which is then a few milligrams still of leftover. So you see here, for this price it makes sense just to use the primary material and not to set up a secondary standard even instead. Right, so that was what I wanted to tell you. I thank you very much for your attention, and if you have questions, please feel free either to send those to me under my email address, christianzeine@lgcstandards.com. You can also send your question to ask us at LGC Standards, as I showed before already. I would be very happy if you can visit our website at pharma.lgcstandards.com. So thank you very much, and have a good day. Bye bye. Thank you.